In this slide, we see a very basic overview of cell metabolism. Basically, we're going to bring in glucose, which is C6H12O6, and then we're going to take that 6-carbon molecule and we're going to snap it in half to create these two 3-carbon molecules called pyruvic acid. Now that process is called glycolysis. Glyco is sugar, lysis is to break apart. So glycolysis or glycolysis is the breaking apart of the sugar. Now when this happens, we're going to create 2 ATPs. Okay, actually we create 4 ATPs. However, if you're going to break something apart, what is it going to take? Like if I take a pencil and hold it in my hand and concentrate real hard, can I break it in half? No. It's going to require energy in order to snap that pencil in half. Okay? So, while we make 4 ATPs during glycolysis, we have to use 2 ATPs to actually snap the sugar. So just like if I bought something for $2 and I sold it for $4, what's my profit? My profit's $2, right? Same thing here. We made 4 ATPs, took 2 ATPs to snap it in half. Our profit is 2 ATPs. Okay? Now, oxygen is required. Okay, if you don't have oxygen, then you go into fermentation or anaerobic respiration. And if we see here, for us, when we go into anaerobic respiration, we're going to create 2 lactic acids, and all the ATPs we've gotten are from glycolysis, and it's going to be those 2 ATPs. Okay? But if we have oxygen present, then that pyruvic acid is going to make its way to the citric acid cycle, also called the Krebs cycle. And again, carbons get exchanged, and we actually wind up with another 2 ATPs. Okay, so still not a lot for our investment in that glucose molecule. But here's where the ATPs really get mass produced. It's going to be in what's called the electron transport chain. And I'm not going to go into a lot of detail about this, but during glycolysis, some electron transporters are made. And during the Krebs or citric acid cycle, a whole lot more of these electron transporters are made. And they're going to transport an electron to that electron transport chain. Okay. And basically what happens here, and again, it's a lot more detailed than what I'm saying, but we're going to take those electrons in the form of hydrogen ions, we're going to pump them up against their concentration gradient. Okay. So if we pump something up against its concentration gradient, what does it want to do? Think about it like this. If we take water and we pump it up into a big water tower, okay, so it's way up there. There's lots of water in this tank. What does it want to do? Well, it wants to leave. And what kind of energy do we have up in that tank? We have potential energy. Now, what if I take the pipe that is coming from that water tower and I put a turbine inside that pipe? Okay, as water is released and goes down through that pipe, it turns that turbine, which might be able to generate some electricity. Okay, I need to patent this. But I don't think they do this, but every town has a water tower. So again, that water running through that turbine is going to generate some electricity. Maybe it's some to help the pump. Maybe it's some just to help light the area, whatever. It generates some energy. Okay. Well, we're transporting these hydrogen ions up against their concentration gradient, and there's only one way down. Just like with the water tower, you have the main line that comes down for the water to go off to the houses. Well, same thing here. We have one way for those electrons to leave, and that is through a channel called ATP synthase. Okay, ATP synthase. And so as the electrons run through this ATP synthase, it generates what we call a proton motive force. Okay, proton motive force. And that proton motive force is just like that turbine spinning and creating a little bit of electricity. This is creating enough force to phosphorylate ADP. ADP, adenosine diphosphate, is an adenosine with two phosphates. In order to make it ATP, adenosine triphosphate, we have to add another phosphate to it, and that takes energy. Okay, remember when ATP snaps in half, it releases energy? Well, in order to put it together, we need to put energy in. And this gives us the energy to take a phosphate, put it onto ADP, and now we have ATP. And so at the end of the electron transport, plus the glycolysis, plus the citric acid cycle, we wind up in aerobic respiration. This is with oxygen. We wind up with 36 to 38 ATPs, 6 carbon dioxide molecules, and 6 water molecules. So this is one of the places where that CO2, that carbon dioxide, comes from that we have to exhale. And then we bring in the oxygen, and it all starts all over again. Okay, so I hope that helped a little bit. It's a lot more complicated than this. Your book will go over it in a lot more detail, but hopefully this gives you the big picture to kind of understand what's going on. Next, we're going to look at protein synthesis. And we'll look at another slide, but I want to point some things out. First of all, we have a strand of DNA. DNA is a double helix. Again, it looks like a ladder that we've twisted. I think I've talked about that already. Remember, we have the A pairs with T, G pairs with C. So adenine and thymine pair together. Guanine and cytosine pair together. Now that's in the DNA strand. Okay, during a process called transcription, we're going to make a single-stranded mRNA strand. That M stands for messenger, so messenger RNA. Well, in RNA, it's a little bit different than DNA. Not only is it single-stranded, but we don't have thymine in RNA. Instead, we have uracil. And so if we're replacing the thymine, what paired with thymine again? Oh yeah, adenine. So if we don't have thymine anymore, we have uracil. So what's uracil going to pair with? Well, it's taking thymine's place, so it's going to pair with adenine. So in the single-stranded messenger RNA, A will pair with U. Okay, so A will pair with U. All right, and then that messenger RNA is going to go to a ribosome where it's going to code for a specific protein. Now, I just want to say, see these little things that are, they're almost T-shaped? Well, this is tRNA, transfer RNA. And these little things sticking out are called anticodons. And again, this could be a uracil, it could be an adenine, it could be a guanine, or it could be a cytosine. Notice I didn't say thymine because, again, it's an RNA, so there's no thymine. So it's going to have a G, C, A, or U. Okay. And that combination, and there are charts. Google these charts, an amino acid chart or a protein synthesis chart. Okay. And it'll tell you what, oh, actually, codon chart is what you want to look up. Codon chart. Because remember I said that these are anticodons? Well, these three bases are called the codon. And so this anticodon has to pair up with this codon. So like if this were an A, adenine, what's this going to be? That's going to be U, uracil. If this is a G, for guanine, then this is going to be cytosine. If this is cytosine, this is going to be guanine. Okay, so these are going to code for a specific amino acid. Okay, and this tRNA is going to hold a specific amino acid. Again, you'll see that on like a codon chart. And then this tells you what we need. So let's go to the next slide here and talk about this. So think about this as doing a recipe, okay? Because, I mean, they call DNA the blueprint of the cell. I think about it more as the recipe book for the cell, okay? And so how are books divided up? Oh yeah, into chapters. Well, DNA has chapters. They're called genes. Genes code for a group of proteins. Now, if you take a cookbook and you want to open up to a specific chapter to make that recipe, what do you have to do? You have to open that book. Well, that's what happens here. We find the recipe for the protein that we want, and we're going to open up the DNA, just like a book. And right here is the recipe for what we want to make. Okay. And so if we're going to write down that recipe, what is that called? Like when like secretaries will write down, what people are saying, or maybe they're listening to, a headset, where there's been dictation and they're writing it down. Well, that's called transcription. Okay. And I don't know how many transcriptionists there are anymore, but that used to be pretty prevalent in offices and such. But that's called transcription. So what we're doing is transcribing or writing down that recipe. Although we're writing it down kind of almost like a mirror image, but since the single-stranded RNA does not have thymine, it has uracil, we're also kind of making it kind of in code. Okay, it's kind of like this. What if you make something and it's this incredible recipe, and I'm like, oh, this is great, can you write it down for me? So you write down the recipe for me, and then I'm like, you know what, I have to take off, but just put it in this envelope. I'll have the messenger come bring it to me. Okay, well, here's our code. Okay, here's our written down recipe. Okay, and our messenger RNA is going to bring me that recipe so that I can make, whatever it is you made. But let's suppose you speak a language that I don't, which would be every language but English. And I do know sign language, but it would be hard to sign a recipe, put it in an envelope, and send it to me. But let's suppose you know a language that I don't, and you write it in your native language. Okay? So messenger comes to me, brings me the recipe. I open it up, and I'm like, oh no, I wanted to make this tonight, surprise my wife. What do I need to do? I need to get it translated. Okay, so if I get it translated, then I can make the recipe. So during translation, this is where the synthesis of a protein is made at a ribosome. So let's take a look at that. So here our messenger RNA showed up at the ribosome. Okay, so here's the kitchen. Although remember I said ribosomes remind me of a stapler where you've got the large subunit. Let me get my arrow working here. The large subunit and the small subunit. And staplers do what? They staple things together. Well, these are going to be stapling together amino acids. And so each of these three codons are going to pair with an anticodon, which codes for a specific amino acid. Okay. So right here, again, let's suppose this is, I don't know, uracil. What's it going to pair with? It's going to pair with adenine. If this is guanine, it's going to pair with cytosine. If this is, I don't know, adenine, then it's going to pair with uracil. Okay, so you got the idea there? So one amino acid comes in, attaches. Now we have an open tRNA binding site for another one. So whatever this code is, we have to match it up to the anticodon. Okay, so this codon has to match up with this anticodon. So the right one comes along from the pool of amino acids and transfer RNAs. It attaches, and now this is where the ribosome is going to staple or attach the two amino acids together. And then that transfer RNA goes away to pick up another amino acid that it's specific for. And then what happens here? Well, we start all over again. Another one comes in, attaches, and then we start to build these long chains, these long amino acid chains. Now the kind of bond that we're hooking these amino acids with are called peptide bonds. And so here's a whole bunch of peptide bonds that are making this chain. So what's another name for a whole bunch? It would be poly, right? Poly means many. And so this is going to be a polypeptide chain. This polypeptide chain then is going to be folded in a very specific way, kind of like origami, right? Folded in a very specific way, and now that becomes a protein. Okay, so I hope all that made sense. It's hard to kind of explain it one-dimensionally here, two-dimensionally here. So go on to Google or onto YouTube and go ahead and look up protein synthesis, and sometimes animations help you, to kind of visualize what I just described. Okay, let's look at the cell cycle. Now the cell cycle is where the cell is going to reproduce itself. Okay, and the different phases it goes through before that actual reproduction. The first phase in cell cycle is going to be called interphase. And this is the phase between cell divisions. And there's actually three phases within interphase. There's the G1 or gap 1 phase. And here it's just routine metabolism. The cell has just split in half. It needs to just metabolize. It needs to grow because obviously if we split in half, we're half the size we started. And now it just needs to kind of grow up. Now in the S phase or synthesis phase, this is where DNA replication takes place. So we're going to take the DNA and make a copy of it. Because what we're working toward is splitting that cell in half. Well, if we don't replicate the DNA, if we split the cell in half, then we only have half the DNA in each half of cell. But if we duplicate it, then we can put half, which would be a full amount, of DNA in one cell, and then the other half, which would be a full amount, in the other cell. And now we have a full amount of DNA in each cell. Okay, so I hope that makes sense. It would be like if, I'm studying for a test and somebody comes up and says, oh, hey, can I borrow your notes? Well, I'm kind of using them, so here, let me make a photocopy of them. I make a photocopy of them. I come over, with the pile. I take half of it, which is a full amount of notes, give it to you. I keep my half, and then we split. We go opposite directions. Well, that's what's going to happen here. The DNA half, which is a full amount, goes to one side. The other half, which is another full amount, goes to the opposite side, and then the cell splits. And then we have two cells. Okay, so S phase is DNA replication. And then we have our G2 or gap 2 phase. And again, that's just routine metabolism. The cell is just hanging out, waiting to split. Okay. Then we have mitosis. Now, mitosis is really splitting of the contents of the nucleus. So we're really talking about what's happening in the nucleus and its contents, which is the DNA. Okay. And the phases for mitosis are PMAT. Okay, PMAT, or prophase, metaphase, anaphase, and telophase. Okay, and I'll show you those in a little bit here. And then we have cytokinesis. Cytokinesis is division of the cell cytoplasm. So this is where the cell, okay, half of the contents are at one side, half of the contents are at the other side, and now it has to pinch in half and pull apart, and now we have two individual cells. That's where cytokinesis comes in. Okay, so looking at mitosis prophase. Now, to me, when I look at it under the microscope, what we're seeing here is the chromatin thickening up into chromosomes. Okay, because remember, chromatin, we can't see that under a regular microscope, under a light microscope. So it needs to condense, and it condenses into those chromosomes, which make them thicker, and then we can see them under the light microscope. And to me, I don't know if we have any fisher people out there. Anybody likes to fish? So I like to fish. If you want to see the latest fish I caught, just check my Facebook page. Caught a nice little over 17-pound salmon a couple of weeks ago from when this is being recorded. And anyway, I like to fish. Now, sometimes when you cast, depending on what kind of reel you have, you might wind up with your line getting all bunched up in a knot. Okay, we call that a bird's nest. Well, to me, this looks like a bird nest of fishing line. Well, I used to watch all these fishing TV shows, and I watch these guys who go on and they've made millions of dollars fishing tournaments. Okay, especially this one guy, Kevin Van Dam, KVD. So again, if you're really heavy into fishing, you might know who I'm talking about. The guy's made millions in tournaments, and that doesn't even come close to what he makes in products and all this other stuff. Well, I fish tournaments as well, but I wind up spending money and not making money. So my point is, what kind of fisherman would you rather be? You'd rather be one of those pro fishermen. So when you see all this stuff that looks like fishing line, don't think of me. Think of the pro fisherman or pro phase. Okay, pro phase. So what's going on here? So this is the beginning of the process of division. Chromosomes condense, as I mentioned, and then the nuclear envelope is going to disappear. Okay, so the nuclear envelope disappears. The centrioles are going to migrate to opposite poles. So here's the centrioles. They're going to migrate to opposite poles of the cell. And what they're going to do is they're going to stretch out these spindle fibers. Remember I said spindle fibers are kind of like zip lines? Okay, so if you've ever been on a zip line, kind of know what I'm talking about. So, so these asters, okay, so that's what this is, aster, and spindle fibers form. The next one is metaphase. Meta means middle. And see how this is lining up in the middle of the cell? So all the chromosomes here are lining up in the middle of the cell. Now half of a chromosome is called a chromatid. Okay, chromatid. Because remember, this is our original DNA. Then we have our copy of DNA. So this right here is double the DNA. Double, double, double. Okay. So the chromosomes are going to line up at the equator of the cell, the metaphase plate. Again, think of meta as middle, with the centrioles at the opposite end of the spindle fibers attached to the centromeres. So now we've got our zip lines. Our chromosomes are attached. Okay. And they're attached at the centromeres. And, you know, they're putting on their little helmets, getting ready to do the zip line and signing the consent form. And so are we ready? So they're going to zip line to opposite ends of the cell. And there we go. See, they're riding the zip lines to each end of the cell. We call this anaphase. Remember, we talked about the meaning of anatomy. Ana means apart. Tomi means to cut, so cut apart. So these are pulling apart, or we can say ana means away from. These are pulling away from the center and going to the opposite ends of the cell. So the centromeres divide. Each chromosome goes from having two sister chromatids to being two separate chromosomes. Sister chromatids. Wasn't that a band in the 70s? Huh. Oh, maybe not. Although if anybody out there is starting a band and can't come up with a name, boom, there, I just gave you one. Sister chromatid. All right. So, and then the spindle fibers contract and the chromosomes are pulled to the opposite pole. Okay. I guess that would be like hooking up to a zip line and then it dragging you to the opposite end. So that's probably more what it's like. Okay, but anyway, anaphase, we're going away from the center to the opposite ends. And then in telophase, or some people pronounce it telophase, but in telophase, the nucleus divides. The chromosomes are at the poles of the cells, and then the nuclear envelope reforms around the two sets of chromosomes. So there we go. We rebuild the nucleus around the chromosomes. And so this is our telophase or telophase. This is what it looks like in a plant. And sometimes it's a little bit easier to actually learn or memorize these from plants, and I'll mention that again just in a little bit here. And then we have cytokinesis, the division of cytoplasm. So in animal cells, which were considered in the animal kingdom, a cleavage furrow forms and separates the daughter cells. And you can see that in this cell right here. And a little bit different than in the plant cell. But you can see this phase taking place in an animal cell and in a plant cell. And so one more recap here. We have this cell that is in interphase, okay, which is the first part of the cell cycle. So again, interphase, it's just going through routine metabolism. There's a point where it replicates its DNA. And so then we get into mitosis itself. And this is where the chromosomes are starting to condense, okay, and the spindle fibers are starting to form. And then we have all the chromosomes lining up in the middle. What's middle again? Middle phase? No, not middle phase, but metaphase. But that's how you can remember that. I think of middle phase. Nope, it's metaphase. And then when they go to the opposite ends of the cell, again, they're going away from the center. What's another word for away? Okay, or apart, that would be ana, so that's the anaphase right here, anaphase. And then we wind up with telophase as the nuclear envelope rebuilds, and we start to get a cleavage furrow, and then we go through cytokinesis. So we're finished with mitosis. Now we're into cytokinesis, where we actually split the cells into two. And, now, is it just me, or does this kind of look like Cookie Monster? Okay, maybe it's just me. I think I need a snack. All right, like I said, it's a lot easier to learn mitosis by looking at a plant cell. Okay, because this is the picture I'm going to be putting on the practical exam, and you need to be able to identify each of these phases of mitosis. Okay, and before you start freaking out, don't worry, I'm going to show you how to memorize them. And you can download your practical study guide if you want to look at this and then label. So the first one, okay, before I go any farther, plant cells are going to be rectangular because they have a cell wall. Animal cells do not have cell walls. Okay, so that's why these look like little bricks stacked up. Okay, because this is, again, the cell wall, and well, it just gives them that particular shape. So here's the nucleus within this one cell. So we look at this. What's it doing? Oh, I don't know. Well, then it's probably interphase if you can't tell. So what is this one? Yes, interphase. How about this one? Interphase. How about this? Interphase. All right, well, anyway. So those are interphase. This is getting kind of boring. So then we come up to this. Look at this. Or even this one. Doesn't that look like tangled up fishing line? And tangled up fishing line, fishing. Oh, those pro fishermen that are making all that money. Okay, so this is what phase? Prophase. Okay. So prophase. Not amateur phase, prophase. Okay. And so then all those chromosomes are going to line up in the middle. Is that middle phase? No, it's metaphase. So meta means middle. So metaphase. And now they're going to start to pull apart and go away from the center. So apart or away is ana. So here is anaphase. Okay, and then we get to this one, and you know what it kind of looks like? Let's see if I can get my pen to come up here. Let's see, I'll just trace it out. And there we go. Okay, not a very good drawing. I was going to draw more to it, but you know what? I think this will be easier. Let me snap my fingers. Ah, there we go. All right. I should have done that with this part too. Well, anyway, so what do we have here? Ah, this is a phone. Wait, not any kind of phone. This is a telephone. See, this is where you put your ear. This is where you talk. This is where you dial. Anybody ever seen one of these? Maybe at a museum or something? Now, see, when I grew up, this is what we had. And I'll tell you, it was not fun. It would take forever to dial a phone number. And then what would always happen, too, is you'd be down to maybe that last number, and maybe it was a 9 or a 0, and you would start to dial, and your finger would slip out, and the dial would go back, and it's like, then you had to hang up and start over again. But anyway, so when you look at this, think about, and I wish I had drawn it a little bit better, but think about a telephone. Okay, so this is telophase. All right, down here, look, another telophase. See how it looks like, well, this one actually looks like a little flip phone, doesn't it? And so what kind of phone would that be? A cell phone. Get it? Cell. We're looking at cells. Okay, never mind. But anyway, so think of this kind of looking like a telephone. So that's telophase. Same thing here. Telophase. Okay, let's see if I can quiz you a little bit and see if you get it right. So I'm going to go out of order though. Okay, so here we go. What's this one? That's interphase. What is this one? Interphase. What is this one? Yep, prophase. What is this one? Telephone phase. Okay, not telephone phase, but what? Telophase. How about this one that's going away from the center? Away or apart. Anaphase. How about this one that is lining up in the middle? Not middle phase, metaphase. And how about this one? Prophase. How about this one? Telophase. How about this one? Metaphase. This one? Anaphase. How about this one? Prophase. It's hard to see, but you can see that it's kind of looking like watered up fishing line. For the test, I'll be more specific though. I'll show you one that looks more like fishing line in there. Okay, one more time. What's this one? Metaphase. This one? Anaphase. Ana away from. This one? Telophase. This one? Telophase. Okay, good. Hopefully you'll do great on the practical exam. Now, going back to mitosis, what we just looked at, the cell is going to duplicate its DNA. It's going to then split in half, and each cell is going to have a full amount of DNA. We call those diploid cells, okay, or 2 lowercase n cells, okay? Now, meiosis is quite a bit different. Meiosis is what takes place in sex cells. And when I say that, I mean like the sperm and the egg. Now, it's going to be very similar to mitosis, except do we want a full amount of chromosomes in the sperm, and do we want a full amount of chromosomes in the egg? No, we don't. So that's not going to work. We want half the amount of chromosomes from the male in the sperm. We want half of the chromosomes in the egg from the female. And when they combine, that'll give us the full amount of chromosomes. Okay? So that process is going to be meiosis. And again, like I said, it's going to operate, especially the first half here, it's going to operate just like mitosis. So we start at prophase, and by the way, let's back up to interphase, which is not showing, because again, that's not part of meiosis, but it's still part of the cell cycle. What happens in the S phase of interphase? Synthesis, right? S is synthesis. So we're synthesizing or replicating the DNA. So this has twice the amount of DNA in it, once again. And it's going to go through what we call prophase 1, metaphase 1, okay, anaphase 1, and telophase 1. No different than mitosis. It splits into two cells that have the full amount of chromosomes. This one has a full amount of chromosomes. This one has a full amount of chromosomes. But here's where the difference is. It's not going to take the time to duplicate the DNA here or here. Okay? It's going to go ahead and go through metaphase 2, anaphase 2, and then telophase 2. So remember mitosis, I said it's kind of like this. I've got a copy of, the notes for the test, my study guide. Somebody comes up and says, oh, hey, can I get a copy of that? Yeah, okay, hold on. Let me run to the copy machine. I make a copy. I give you a copy. I get a copy. And then we go our separate ways. Both of us have a full copy of the exam. How many of us have it? 2 of us have it. At the end of mitosis, you wind up with 2 cells, 2 daughter cells, that have a full amount of chromosomes in them. Meiosis would be more like, I've made a copy for the other person. I give it to them. There's 2 of us that have a full amount of the study guide, okay? And then 2 more people walk up and say, oh, hey, I didn't get a study guide. Can we borrow your study guide? Look at my watch. Okay, we don't have time to make a photocopy. So I tear the paper in half, give that person half, I keep half. The other person that had the full amount, same thing. They rip theirs in half. They give them half. They keep half. How many of us have copies of the notes? That would be 4 of us. But do we have full copies of the notes? No. Each person has a half a copy of the notes. Same thing with meiosis. Theoretically, at the end of meiosis, we wind up with 4 daughter cells. Each cell has half the amount of chromosomes. Okay? I say theoretically because that doesn't really happen when we talk about the egg, but that's a topic for next semester. And so since these cells have half the amount of chromosomes, are we going to call them haploid? No, pretty close though, but that's how I remember it. It's not haploid, it's haploid. Okay? Haploid. So if you're haploid and you know it, clap your hands. Okay, I know that was dumb, but hopefully it helps you remember.